Hello, Professor. My name is Anyadita Udaya, and I'm going to talk about a paper on ICOS-expressing CAR-T cells mediating durable eradication of triple-negative breast cancer and metastasis. First of all, triple-negative breast cancer of TNBC is a type of breast cancer that lacks estrogen, progesterone, and HER2 receptors, accounting for all 15 to 20% of all breast cancer cases. It has high risk of recurrence and poor prognosis. So, existing treatments such as tamoxifen and trastuzumab are limited in its effectiveness, particularly against metastasis and solid tumors because of its tumor heterogeneity, immunosuppressive microenvironments, and CAR-T cell exhaustions, as well as poor tumor infiltration by CAR-T cells. And to address this, the study hypothesized that integrating the inducible co-stimulator ICOS to CAR-T cells targeting the B7H3 can enhance anti-tumor efficacy, persistence, and cytokine production, so it will improve outcomes for both primary tumors and metastatic sites. Here, they design the CAR-T cells that target a specific protein B7H3 found on TNBC cells and made the CAR-T cell express ICOS. B7H3 is highly expressed tumor antigen in TNBC, but not as much in normal tissues, making it as suitable inducer for ICOS to be expressed. In the design of ICOS CAR-T cells, the extracellular domain contains a single chain variable fragment, or SCFV, that binds specifically to the B7H3 antigen. And the transmembrane domain anchors to the receptor of the T cell membrane, ensuring its stability. And inside the cell, the intracellular signaling domain integrates ICOS alongside standard signaling components to amplify activations. So, in the mechanisms, the SCFE binds to the B7H3, and this will activate CAR-T cell and triggers the cytotoxic response and releases the ICOS CAR-T cells, which will increase the cytokine and granule productions. This allows the recruitment of other immune cells and amplify the response and kill the tumor cells directly. From the in vitro and in vivo experiments, we can see that ICOS CAR-T cells showed a significant cytotoxicity against the NBC cell lines. In the E graph, we can see that ICOS CAR-T cells achieve over 60% of tumor cell killing, significantly outperforming standard CAR-T cells, which achieve approximately around 40%. So, in this figure of F, it highlights that ICOS CAR-T cells release higher levels of cytokines such as IFN gamma and TFN alpha, which are the indicators of robust immune activations. And these results validate the superior functionality of ICOS CAR-T cells in controlled lab conditions. G and H also support this finding, showing high levels of cytokines secretion in vitro. And lastly, the I and J show the TNF alpha and IFN gamma cytokines kinetics over time. And in the in vivo testing, mice were injected with the NBC cells to induce metastatic tumors, followed by treatments with ICOS CAR-T cells, conventional CAR-T cells, or no treatment as control. Then tumor burden and survival rates were monitored over time. As demonstrated here, ICOS CAR-T cells significantly reduced tumor size and weight in mice injected with the TNBC cells compared to control groups. This further showed by B and E that is also successful in eliminating metastatic tumors, especially in major organs. Lastly, I would also like to discuss the role of ICOS-L as natural ligand biomarker. Backed up by these tests, A2I reveals that tumor with higher ICOS-L expression showed better response to ICOS CAR-T therapy, demonstrating increased tumor killing and cytokine production. These findings suggest that the ligand could guide the patient selection for therapy, which maximizes outcome for individual with ICOS-L positive tumor. Like, the more ligand it has, the more effective the therapy will be. So to sum it up, here are the key points from this presentation. And in the future, we can develop this field by ensuring the validation of ICOS CAR-T therapy in human clinical trials, expansion of ICOS-based CAR-T cells designs for other solid tumors, and development of combination strategies to upregulate ICOS-L in tumors with low expressions. That's all for me. Thank you for your attention.